which can be compared. The importance of this qualitative data is for classifying objects that can see patterns, frequencies, or associations among groups. Okay, so that's the particular importance of ordinal data. Okay, so we just move right along. So basically, you want to talk, if supposing you are asked a question on the two broad categories of data, how do you go about answering them? How you answer them is key. So if you are asked the broad categories, you know that you are talking about qualitative and quantitative, and you want to give me their subcategories, because that's how you be able to get full marks. All right, you want to give me their subcategories, you want to give me examples of how you want to give me examples of how these data are grouped for qualitative data. Okay, you want to specify that they are categorical, then you want to say what is their major attributes. Like we said, they are important for classifying objects into categories which can be compared to see patterns, frequencies, and associations among groups. So if you say all of that, you would have significantly say something about what qualitative data. And the next one you want to move into is what quantitative data. Now this quantitative, just like qualitative that is also known as categorical, this qualitative data is also known as numerical data. It's also known as numerical data, and they are data that refers to values, numbers, figures that can be measured or counted. That is very key. Examples of quantitative data in biology, like we saw, we have heights of plants which can be measured in centimeter. Then we have body temperature, can be measured in degrees centigrade. So you have specific numbers around quantitative data, and you have things like number of offspring produced by an organism. So that's for quantitative data. And the next aspect of that quantitative data that you want to tell me is the discrete and continuous. So you know that quantitative data has discrete and continuous, two categories or subcategories in it. And for qualitative data, their subcategories is what nominal and what ordinal, and this can be referred to as the types of data under what qualitative data. That's nominal and ordinal, and the types under quantitative data is what discrete and what continuous data. So you want to say what discrete data is, and we said that discrete data represent numbers or whole numbers. These values are separate and cannot be subdivided. Okay, they are separate and they cannot be subdivided. While continuous data represent measurements that can take up any value within a range. Okay, they can take up any value within a range and they can be subdivided. So why your discrete cannot be subdivided? Okay, they cannot be subdivided. Your continuous data can be subdivided. They can be subdivided. So you should be writing like pari passu what this is doing and what this is doing that the first is not doing. That's how your communication in answering the question should go. Alright, so we move to the next thing we saw, and that is importance of quantitative data. So understand the categories in which we took all this stuff, both quantitative and qualitative. We look at what they are generally. We look at the alternative name for them, categorical and nominal. We look at the examples that are within these categories, and we also saw the types of types, there are subgroups under this category. After you've looked at their subgroup, the next thing is to know is their what importance. Okay, of what importance are they? All right, so importance include that they allow researchers to measure variables, okay, accurately, and perform calculations to test hypotheses. That's for qualitative data, and that is a major importance of quantitative data. So you want to look at some statistical tools, but in quantitative data we also have their types of quantitative data. We saw discrete, we saw continuous. Okay, we've looked at what discrete and continuous are. You should know them. Then the examples of discrete, the examples of what continuous, their characteristics. All of these are key. Okay, so then we now look at the statistical tools for analysis of discrete data. The statistical tools, we saw that for discrete data we have frequency counts. You can count them. You have things, statistical tools. This is very key because it's the core for knowing what statistical analysis to apply. So for statistical tools, you saw that they have frequency counts, they have bar charts, chi-square. These are statistical tools that can be used for what qualitative analysis. Okay, and you also have for quantitative analysis things like histogram, okay, histogram, regression analysis, t-test, ANOVA, analysis of variance. So that's basically the aspects that we looked at. So I just want to take one of the questions asked here. He said recap the first part of the word lecture, which was what? Okay, which was not streamed. Okay, so the first part of the lecture is basically the definition of biostatistics that I was talking to you about. That is the application of statistical method to biological research. Then we saw that the different aspects of that biostatistics is that they collect data, they analyze data, and they interpret data. And all three of these are key for biological field research. So you must definitely have to collect data, analyze the data, and what interpret the data. And in doing this, you can also apply this in what medicines. You can apply it in ecology, genetics, and what public health and more. Okay, so These are examples of disciplines that they can be applied in. Okay, so that's actually the beginning of this part of the lecture. Then in the next slide or page, we saw the importance. So yeah, I think I started from here, but I didn't do much here. Okay, I said the importance of biostatistics in biological research. Here you are looking at the general importance of biostatistics. Okay, and this is not related to your test questions, because some of you actually wrote this as answer when I said importance of uh, statistics data to biomedical research. Okay, there was this confusion. So this is generally both biological research, biomedical, environmental. This applies. It reduces bias, minimizes errors, ensure reliable results. Of course, designing effective experiments. If you are in class, I dwell much on designing effective experiments. It guides decision making, and it understands biological patterns and trends. All right, so that's pretty much the first part before we went into types of data that we just concluded. So we have concluded types of data like this. So if you have any questions or areas that you are not clear with in how to tackle questions around this aspect, please drop it in the comment section, so that as I move right along, I'll stop to take your questions. So the person that asked that question, if you are okay with the question, please also drop it in the comment section so that I know we are proceeding. Okay, so now we saw bioassay. Bioassay here is key. All right, and is simply an estimation or determination of concentration or potency of physical, chemical, or biological agents by means of measuring and comparing magnitude of the response of a living organism. Okay, so the bioassay have different types. So important that you know the general definition of bioassay. And bioassay can be said to be based upon the use of what biological active substance. So biological research generally are referred to as assay. When you're in the lab, you are always carrying out research or assaying for one aspect of your research work or the other. So they are generally called bioassay. So you know the definition and know it clearly. Alright? Then the next aspect of the bioassay, it also now relates to where we are coming from, which is your qualitative assay and quantitative assay. So qualitative and quantitative here are pretty narrowed down to bioassay, and important you know the difference. You say these assays do not present any statistical analysis. That's for qualitative assay. Like I said, they just want to know if whatever you are measuring, for instance, you are doing a phytochemical research, if whatever you are measuring, they just want to know that, oh, is present, flavonoid is present, phenol is absent, and you indicate those by plus or minus. All right, so but for quantitative, you want to know the amount, you want to know how many milligrams per liter of it is present, or the percentage present, or the concentration moles per liter of each present, depending on your research that you are carrying out. So quantitative assay here provides numerical assessments of some properties of the material to be assayed, and they pose what statistical problem. So what actually leads to the application of statistics in bioassay is the application of quantitative assay. With qualitative assay, you might not, in fact, I don't think you need any statistical tool to assay that. You just need a plus or minus, a color change, to know that what you are saying is either present or absent. But when you are not carrying out quantitative assay, you are talking about the amounts, you are talking about optical readings, OD readings, and you are talking about calculations. You are talking about taking your sample spectrophotometric readings in triplicates. You are talking about taking the average. So at the end of the day, they provide numerical assessments of some properties of the material to be assayed. And with that, when you have those numbers, that is where statistics now come in. You need statistics right there. You need to know the right statistical tool to apply to whatever data you have collected in form of readings or measurements or whatever. Now, those statistical measurements is now applied to it. So characteristics of bioassay includes that it has active principle to be assayed and should be the same measured response in all biological species. Then the method selected should be reliable, it should be sensitive, should be reproducible. Very important for characteristics of what? Bioassay. Okay, so if you are asked about the characteristics, the first thing is that you want to know about the active principle to be assayed should show the same measured response in all biological species. So it should be reliable. Number two, whatever method you apply in biological research should be reliable, it should be sensitive and reproducible, and of course it should minimize error. Remember the last class we talked about bias and error. The whole essence is that in any biological research, as much as possible, excuse me, you want to minimize error due to biological variations or methodology. It could also be due to the experimental design.
Someone asks, qualitative or quantitative assay.
Write it. Just let me write it. Okay, so it could also be due to experimental design. Remember experimental design we talked about in class. Okay, so that's number two characteristics. Then we also have some more. Let's see the next slide. Okay, still on characteristics of bioassay, we said that the reference standard excuse me. We said that the reference standard and test sample should have the same biological effect and mode of action. That's another key characteristics. And we said bioassay might measure a different aspect of the same substance. For instance, you take an experiment that you are using the leaves to measure some phytochemical parameters. Okay, now you use the leaf, same plant, let's say bitter leaf. Okay, you use the leaf for this set of experiment, you measure the same parameters, the same phenolic, flavonoid, alkaloids, okay, saponins and all of that, compared into the chemical assay. You also want to take the same, those parameters, and look at those components in the roots of the same bitter leaf or in the stem of the same bitter leaf. Amazingly, all of them will give you different results. So another characteristic is that activity assayed should be the activity of interest. So you don't want to look at everything. You want to narrow down your research to what you are interested in. Like I told you in class, in carrying out my research for bitter leaf, what I was interested in is the genetic composition of bitter leaf. I was not too interested in phytochemicals or whatever it is that have been researched before. But as soon as I knew that the genetic aspect has not been explored, that is my interest. And every design and every measurement or research parameters or activity or assay that I carried out was geared towards my interest, which is the genetic component of bitter leaf. And that's what you should do when even applying in your research. You know, you are projects and student now, and you are going into research, so all of this should apply. Alright, so we go into types of bioassay. So I have a question here. If the qualitative and quantitative, sorry, is the qualitative and quantitative, are they types of bioassay? Is that your question? Okay, I said qualitative and quantitative from the beginning are two broad categories in which biological research is conducted. Now, when we came to bioassay, in bioassay, if I could quickly recap, are you just joining in? This person asking this question. In bioassay, I said, though we are coming from quantitative analysis and data, actually, the type of data to be assayed, quantitative and qualitative. But for bioassay specifically, we also have qualitative assay and quantitative assay. Do not miss the two. The two has their category. One is a broad general data, and it's called qualitative data. In this bioassay, it is called qualitative assay, narrowing down to the assay itself. Okay, I hope I have answered that person's question. Then the next set of things we want to look at is types of bioassay. This is key. There are three main types of bioassay. We have the direct assay, the indirect assay, and here it has two indirect assay. So if I was to do it and separate it or put it the same, it could be like this. Okay, so we have one indirect assay that is focused on the quantitative response, while the other indirect assay is based on the quanta response. So please note that it's not two types of bioassay, it's three. One direct assay, two indirect, and the two indirect you should know. It should be based on the quantitative response, and the other one should be based on the quanta response. So let's look at what the direct assay signifies. Is doses of the standard and test preparation that are sufficient to produce a specified response and can be directly measured. So the key word here is that it can be directly measured. So if it can be directly measured, you can as well guess right that the indirect assay would be indirectly measured. Okay, so we have indirect assay that is based on what? Quantitative response, and it is the relationship between the dose and response of each preparation. When we say preparation here, we are talking about preparation for your research, which is your bioassay now. So it is first ascertained or determined, then the dose corresponding to a given response is obtained from relation of each preparation separately. So that's the first indirect method based on what? Quantitative response. Then the indirect assay based on quanta, which is all or none, is concentration of known. So quanta, this is basically based on calculating formula, and you will see this a lot in bioassay. You will see this a lot in bioassay, where you have concentration of unknown is equal to dose of standard divided by the dose of the test times the concentration of standard. So most times after you have carried out your research and you have your OD readings, you will need to now convert those readings to concentrations. And it's from these concentrations that you can now even apply statistical methods to it, such that those statistical methods can now lead to a proper interpretation of your results. So most times you'll be needing this formula here. So if this formula is the indirect assay that is based on what? Quanta method or quanta response. So this is it. I'm going to our next page. So we have advantages of bioassay. Advantages of bioassay. So very key. One, you should know them. It helps in determining the sensitivity of the tissue by plotting a concentration response curve with the known against unknown. This concentration response curve is usually with the known against unknown. This also helps in determining the potency of the sample. This is a key advantage right here. It helps in determining the potency of the sample. Then two, second advantage, it is specially used to standardize drugs, vaccines, toxins, poisons, disinfections, antiseptics, etc. They are all used over biological systems in one form or another. They also help to determine the specificity of a compound that is to be used. So you want to know how specific in reaction that compound is. Still on the advantages, we have the disadvantages. Okay, so the disadvantage of bioassay, this includes dosage timing may vary in the mode of application of the drug. Then you also have it is less accurate, more time consuming, and more troublesome. So bioassay is good, but it has this disadvantage. Okay, so its exact match of the response may not be available, and this could be problematic. Okay, so the quantitative difference between standard and test may not obtain any biological variation. All right, then of course sensitivity of the tissue changes with respect to time. So as soon as you carry out your OD, the readings you take over time changes. So as soon as you have, maybe they say take this OD reading at 30 minutes time, it is very important that you take it at 30 minutes time. If you leave it for 1 hour, you already have alteration in your result, because the readings that will come out at 30 minutes time is totally different from that of 1 hour or above. Alright, so that's the disadvantage, and it's key to note. Okay, I have a question here. Does these types of bioassay have preferred statistical method to employ? Of course, the type of bioassay, the results you obtain from each of them is what we inform you on what statistical method to employ. But basically, those statistical methods will not vary from your statistical data. Of course, if you do bioassay and you do quantitative, what you will have is quantitative data. Okay, so based on the type of research that is conducted or carried out, you will now know from those type of statistical methods under quantitative data, you will now know which one to apply based on your results and your numbers. Same thing if it's qualitative data, you will now know what type of statistical method to employ. So this aspect actually goes pari passu with quantitative data and qualitative data. They complement each other. So if you have carried your bioassay to the end and you now have your data, the next thing is to make reference to qualitative data and statistical methods that can be employed. So when you look at it critically, you can now know that, oh, for this type of my research, this method of this qualitative data should apply. Okay, so I hope I've answered that question. Thank you for making this class very lively. You are not sleeping and leaving me. So we have application of bioassay. Application of bioassay. That is another key thing to note in bioassay, how it should be applied. Like this is somehow related to the questions and the question I just answered and explained. So in what area of research would bioassay be applied? Here we say number one, establishing regulatory requirements for water quality. So it can be applied in water assay. If you have anything to do with testing water, knowing the quality and safety and health and risk in a particular water taken from a particular sample source, then application is a bioassay and it requires for water quality. Then you have ecological monitoring and sewage discharge. Then you also have environmental impact assessments of new technical technologies, treatments, facilities, reconstruction, economic project, modernization of national economic project. Then you have determination of drug potency, dosage, side effects. That's for biomedical sciences. Then you also have for environmental, like monitoring of water bodies, particularly in the area of what? Human exposure. These are areas in which bioassay can be applied, and it's important that you know this application because this is what the course is about, applying biostatistics to what? Biological research. So we have importance of statistics in biology and medical sciences. This is actually where your test question came from. So if you don't know the answer now, you are seeing the answer. So it's not the first general importance of biostatistics in general in relating to all biological research. Now this is narrowed down to medical sciences, also known as biomedical sciences. So we are looking at the importance of these statistics in biology and what? Medical research, and that could also be known as what? Biomedical what? Sciences. So number one importance is that it helps in research planning. It also helps in what? Sampling techniques, especially experimental design. We'll be looking at sampling techniques shortly. It also helps with data analysis, research what? Conclusion. Then we also have medical sciences, statistical tools for the collection, presentation, and analysis of observed factual data relating to the causes and incidence of what? Disease. This is exceptionally narrowed again to what? Medicine or medical sciences. So with assay or biostatistics in biological research, you could get some research conclusion, of course do data analysis, sampling techniques, and research planning. So these are the importance. Alright, so we're moving on to the next one we looked at. Okay. So in continuation, we have factual data, pulse rate, body temperature, blood pressure, heartbeat, weight. So all of this in relation to that medical sciences, we said that it's when dealing with that last point of the application, you are talking seriously now and more specifically about what factual data, and it helps doctors to give proper diagnosis of the disease. Okay, then pulse rate, body temperature, body pressure, heartbeat, weight. These are like your vitals that you take when you get to what, the hospital, before seeing the doctor. And patients that visit the hospital with different type of ailments undergo this factual data. We also have statistics that can be used for studying of heartbeats through what, electrocardiogram, which is ECG. When you have things that is related to heart attack, heart rate, my heart beating fast, my heart peppering me, these are the type of statistical um, tools that you use in biomedicine. So statistics can also use the test of what, significance in biomedicine. So one major test that is used for biomedicine or biomedical research is what, a test of student T test, which is what, test of significance. All right. So we look at types of biological and medical data. There are different types for biomedicine, and that includes qualitative type of data. Are you seeing it relating to the first that we already looked at? And in this qualitative is exactly uh, what we saw. It said it uses nominal and ordinal scales. All right, and data on reported cases and deaths from notifiable disease, morbidity data, vital statistics on live births, stillbirths, and maternal births. These are aspects in which you ought to look at critically as a biochemist during your practical aspects or the statistical part of the course. Okay, if it was Dr. Akala Gboro that took you these aspects, you will actually go for a field work in getting these vitals. I don't know if the person that took you now did all of that. Okay, so you also have data on infectious disease and communicable disease. These are types of what, biology and biological and medical data. Oh, thank you. All right. Just hold on, I'm trying to get some things together. Okay, so when looking at, like in the previous page where we saw applications of bioassay, the next, this slide in particular is also looking at that, data in communicable activities, data in alright, I'm coming. Okay, so this under types of biological and medical data. So we have 5 here on the list, and right here we have the others. So let me bring it down. So we also have data on immunization activities, health manpower distribution data. All of these data are available in health centers, immunization centers, or hospitals. You get all of these data. Okay, so another aspect of this biomedical data is the sources of biological and medical data. The major source of biological and medical data is from what? The Federal Ministry of Health. Okay, then the principal source of biological and medical data is the National Bureau of Statistics. We also have the Nigerian Medical Council Association as one of the major sources of what? Biological data. Then we have the Federal Ministry of Health, which obtains routine medical statistics from the following sources. Okay, from the following sources they obtain: from State Ministry of Health, teaching hospitals, specialist hospitals, primary health care, like I said, zonal health boards, local government for routine vital statistics. These are things that you ought to go and get and analyze as your practical session of this course. Then you have National Populations Commission, State Ministry of Finance, then Office of the Accountant General of the Federation for Public Finance and Finance Statistics on the what sector. So these sets here are actually subgroup under what the types, the sources of biological and medical research, and those subgroups are specifically under this point number 4, where you have the point number 4 where you have Federal Ministry of Health that obtain routine medical statistics. So those routine medical statistics has the following sources, and that is what we have stated on this slide. Alright, so we'll just move right along. And we have problems associated with collections of biological and medical data. We have untimeliness, reliability, incomplete, uncoordinated. So most times biological and medical data suffer from this kind of problems and challenge, and they lack proper communication between users and producers of biological and medical statistics. They also lack cooperative attitudes on the part of respondents, and they lack trained personnel. So these are some of the problems that are associated with biological and medical research. So the ways of tackling this problem: how do you solve the problem? Number 1, government and other stakeholders should put what efforts to improve on the management of biological and medical statistics, especially in Nigeria. Okay, so we have computerized database of biological and medical statistics, and medical statistics should be established. Then we also see that there is a need to monitor the management of database to ensure the production of reliable statistics on this critical sector. So these are ways you can tackle these problems associated with biological and medical research. So before I go on, I just want to ask, is everyone following? Are you hearing me clearly? Then again, what time is the exam today? What time? Please, you can type that in. I just want to know the time so that I know how to just summarize and let you go and rest. So we also have types of variability associated with biomedical data. we have three types of variability. That's variables that you can use for biomedical data. You can use natural and random variation, you can use systematic error, and assignable causes. Remember when I taught you in class about sampling methods? Okay, these are type of variables you can use to what for sampling method associated to um, biomedical data. We have natural or random variations, we have systematic errors, we have assignable causes. So systematic errors include personal errors or mistakes. Here you are beginning to see bias and what errors, and that's a topic of its own. I think the last topic we looked at before we wind up the course. By the way, I gave your class an assignment. In the last class I came, I met very few persons a day to your election. I gave your class an assignment that you ought to submit. The reason for that assignment is born out of the fact that you probably did not do very well with your continuous assessment test that was conducted. So I was looking for a way to make up that. So if you did not submit that assignment, you may not be doing yourself any good. I just thought to bring that out. I've not asked for this result, but I'll collect all of it tomorrow. So if you have not submitted, make sure that you do that. Do I have the time for the exam now?
11:30.
11:30 this morning. All right. So we have another type of variation and biomedical research, such as the weight and height of identical twins, increase in body weight in twin pigs that are given the same diet or same management therapy. So all of these are what types of variables in what biological and medical research. Okay, so we have total variation. Total variation in any set of measurements can therefore be written as what? Total variation is equal to assignable causes plus bias plus random error. I think this is where we ended the class at biological and medical research. Huh? AOC. Is the AOC we are talking about? So you think I came here to give you your exam question? But if you are wise, your exam question is being discussed. So you better stay tuned. Some of your questions have been discussed already. So if you are not listening, when you see it, maybe you remember. So this is where we wrap up biological and medical research, and the next set of lectures we moved up to levels of what? Measurements. Okay, so I think this year in class, this is where we started off from, levels of what? Measurements. And of course this I will just run through because you were in class with me for this. So you want to know the levels of measurements, and what is essential. Why is essential? Why is it essential? It's essential for three main things. Okay, and the three main things are: one, to organize, to analyze and interpret data. And that is actually pretty much what biostatistics do in general. So this is key: analyze, interpret and organize data. I would say there are four primary levels of what? Measurement. We have ordinal, nominal, interval and ratio measurement. All these differ in their application in statistical tools. Alright, so you need to know these levels of measurements and know their examples. All right, you know their examples, you know their um, characteristics, which we discussed in class. We discussed much more than that, but um, I'm trying to speed up now so that we could wrap up. So we have nominal level of measurements. You want to know what they are about, how they are classified. Nominal major levels, most basic. They are the most basic type of level of measurement, and they classifies
So
data.
now this class also cover representative sample data collection tools we give for that for
Knows. I heard somebody is asking about if the class is covering representative sample. No, I just talked about the assignment. That is the assignment. That aspect of the course is used for continuous assessment. Okay, and I can categorically tell you that you may not see that in your exam. So just make sure you submit the assignment as expected so that you don't lose the mark. All right, so you are looking for AOC, you are getting it now. Some people are sleeping. All right, so no inherent order or ranking in nominal measurements. They are qualitative and categorical in application. So you can see that even the levels of measurement are in alignment with the types of data, quantitative and what qualitative. So if you understand that aspect very well, this is pretty walkover. They represent groups or labels without any numerical means. So we now look at features and what characteristics of nominal level of what measurements. So this is key. All right, I said you should know their examples, you should know their features. Okay, so here we have no order, no arithmetic operation, no mathematical calculations are what applied. These are what their characteristics or features.
the money asking if the group number is composite for that assignment.
Sorry,
If the group number is composite for the assignment.
I'll answer that later. Let me finish. Just let me note it. So for the features, we said no order, no arithmetic operations, they are mutually what exclusive. Examples of each of these levels is the species of the animals. For example, dog, rabbit, lions. You have blood types A, B, AB, O, eye color, blue, green, brown. These are examples. They show categories and what attributes. Okay, then the statistical tools that are used for this type of measurement include pie charts, frequency counts, and tabular data, etc. All right, so when you know each of the levels of measurement, you should know examples of each level that you mentioned. Okay, and you should know characteristics of each level. Okay, I taught this extensive in class, so we'll just move right along. You could also listen to the
When we say
listen to the YouTube video to further concentrate on this if you were not in class.
okay, now our class does not have questions for the assignment.
Sorry?
When we say now our class does not have questions for the assignment.
Okay, if that is Ken Baukem that do not have the question for the assignment, please talk to your colleague in biochemistry to give the questions. I'm not sure I have the exact questions I asked any longer in my head right now. So please ask from your colleague in pure biochemistry to give you the questions. Number 2 level of measurement is the ordinal level. And in the ordinal level, you will say that this type of level organizes data into meaningful order or what ranking. So they have categories that have meaningful order or ranking. and they have features and characteristics that includes order of category on equal interval comparative. They are comparative only. Examples of this ordinal data is disease severity, socioeconomic status, pain scale, mild or moderate. Okay, and they are used in biological and medical research. The statistical tools that can be used or tests includes Mann-Whitney U test, Crystal Wallis test. All of these are used for the analysis of what ordinal data. And we also have things like the bar chart distribution of virtual representation of what data. They are all used for the ordinal data. And the next is what we saw is the interval level of what measurements. The interval level of measurement includes data with ordered categories. Okay, data with ordered categories and equal intervals between values. So you have the characteristics, order with equal intervals, no true zero. They are quantitative in nature. That means they support mathematical operations like addition, subtractions, but they do not support multiplication and what divisions. Example is temperature, pH level, pH scale, calendar date. All of this shows distinct or meaningful difference between years. So you can always go back to these lectures to see anyone that you are not clear with, so that we'll move very fast. So the last of the types is ratio level of measurement, and this represents the highest level that encompasses all features of the previous levels. That means every feature of the other three levels are in this ratio level, and that includes the fullest range of mathematical. It includes ordered equal interval and is a true 0 point, and it allows the fullest range of what mathematical operations, including comparisons of what absolute magnitude. So features and characteristics, it has true 0, and that 0 means absence of all attributes. I think I dealt extensively in class for this. Okay, and I'm sure the YouTube video for this lecture is up already. If it's not up, it will be up before morning. So after this class, you could just have a nap and sleep, and in the morning look at all of this again, so that we move swiftly. So the next thing we want to look at is the importance of these levels of what measurements, and why are they important in biostatistics. Alright, so it informs the selection of statistical method and ensure accurate and valid data analysis. Example is nominal and ordinal data that requires non-parametric tests to analyze patterns or relationship. Then we also have interval and ratio data that has been quantitative and allow parametric tests. So one allows non-parametric and the other allows parametric. So we got into sampling methods. Alright, and sampling method, we saw the different types of sampling methods, and we said that sampling method is used to select a subset of individual. The word subset is key here, of individuals or elements from a larger population for what study. I think I dwelt a lot on sampling methods, and we said that they are basically what 4 different methods of what sampling methods. We have the random sampling methods, systematic sampling methods, stratified, and what cluster. Okay, so you should know the sampling methods and you should know the details that follows. Alright, so we now got into what explaining each of the sampling methods. So we have simple random sampling method. That's for one of the sampling method. Here you have excuse me. So the random sampling method, which is also known as the simple random sampling methods, right here, is used where individual or elements in the population has equal chance of being selected. Then it is applied when seeking generalizable results. Very important. It is reliable and is what the most reliable form. So each one has its key. Generalizable result, sampling method is the most reliable form. Then we have unbiased in its operation, which is what we want to do. We want to reduce bias and errors as much as possible in research. It works by considering the following. Researchers create a list of the entire population. All of these are in your course material and you should study them. So I just want to move faster than I'm doing right now. So what the research should do. The research will do the following under random sampling method. It will list the plants and all of that. Then it's important you know the advantages of each one, reduces bias and all of that. The exam is not going to be too difficult, I can assure you, not after this class. So it's ID and time resources. I think these are the applications or advantages. So I think that is basically it, and we moved on to seeing bias and errors. So just like we saw for sampling method, the same pattern we had for systematic method, stratified method and cluster method. So I advise you to please look at all of that. Okay, they are in the YouTube channel lectures. If they are not there, somebody will not sleep tonight. You have to upload all of them for you, so that in case you want to just run through the lectures, you could do that. Okay, so bias and errors is what I took in class. Okay, and that could be a bonus. So for bias and errors, you should read it very well and know how to explain it with respect to biological data collection. Then also know the different types of bias and errors. Remember, there are many types, like up to 8 types, even publication bias, experimental design bias, measurement bias, just name it. So each of them, know what is playing in it. And with that, I close this online exam revision. Thank you. If you have questions, please do ask now. Okay, I hear somebody ask if the assignment submission should include the material number. Is this somebody that is asking that question? So I'd like to ask the person, what do you think? What do you think it should be? Put it in the comment section. Is there any question? Please collect my question for me so that we'll go. Do now. So in the absence of no other question, Can we call this class closed?
Somebody said, wait, you will also do a YouTube video for us in biotechnology and genetic engineering. Should we also be expecting like you said?
Yes, for biotechnology and genetic engineering, definitely you will get your videos. That is about your last exam. Can somebody just type what date is that exam? Okay, so I've been pretty busy, otherwise the video should have been up on the YouTube channel, and we are having some serious network issues around where I am, but I can assure you it will be up, not by weekend, but let me know when that exam should be. When is biotechnology and genetic engineering exam, please?
Please do a recap on bias and error.
We should do a recap on bias and error.
They say 5th of March.
5th of March. That's the last day of your exam, right? Is that correct? Are they answering me at all?
They say 5th of March. They don't know if the guy is allowed. Somebody is begging to do a recap on bias and error.
You said?
Somebody is begging for a recap on bias and error. 5th of March is next week Thursday.
Okay, I hear somebody is asking for a recap on bias and error. Yeah, I'm tired though.
Recap the bias and error.
Okay, so let me quickly do that recap. I've closed my whiteboard though.
I think guys will have your 5th of March.
It did not open again. Sir? Hmm? No. There are many. Okay, so I want to before I go into is my phone on? Is my SIM in my phone? So I can access my WhatsApp now. Okay. Because my SIM is not in my phone. It's in my phone. Where's the phone? Give me now. Okay. I want to take like a quick attendance. So please, if you are here from biochemistry class, okay, find a way of identifying yourself. Just put your name on the attendance. I want to know how many people are here, because now they are telling me to recap bias and error. That's how they did not attend lectures that time. It cannot open. Okay. Alright, so
When you say like the bias and error, are we supposed to discuss the type and causes of each type?
if you see discuss the type and causes of each type, you will discuss it now. These people, you want me to read out your examination question for you here? I don't understand. Are we supposed to discuss? I should not say yes. If you see discuss the types and whatever, you discuss it. If you do not see it, you answer accordingly. Okay, so what did we say bias and error is in biological research? Okay, I've not even done that video myself. So we said bias and error in biological data collection can significantly impact accuracy, reliability, and validity. Yes. So we say bias bias and errors. So what is this about? This can significantly impact the following. One, it can impact accuracy. It can impact reliability. And it can impact validity. And all of these affect what? Research finding. So it can impact accuracy of research finding, reliability of research finding, and validity of what? Research findings, which is basically your data that you analyze with statistical method. So in biology, these biases and errors can stem from various sources related to either sampling method, experimental design, research influence, and what? Data analysis. Therefore, understanding the sources of bias and errors helps, this is key, understanding the sources of bias and errors helps researchers takes preventive steps to minimize inaccuracies and ensure the data accuracy. Ensure the data accurately represents the phenomenon being studied. So this here that I just stated could be said to be what? The general significance of understanding bias and errors. Okay? So you could say the general what? Importance. General importance of what? Bias and errors. Okay, so that is key. And when you know this importance what bias and error, is key to know what bias and error is in itself and what is his focus. His focus is what? To significantly impact. You want your research findings to be accurate. You want it to be valid. That means what you are saying is correct, is valid, is in alignment with today's research and precedence. Okay? So you want it to be reliable. Okay? So you want it to be reliable. So understanding that what the bias and error in general is key. Alright? Then the next thing is that the general importance of the bias and error in itself is that the sources of the bias and errors will help researchers to take what? Preventive step. That is key. If you know what could lead to bias and error, you don't want it. You want to avoid it. You want to minimize it. Okay? You want to prevent it as much as possible to minimize inaccuracies, that's incorrectness or false values, false result, and ensure that the data accurately represents whatever it is that you are studying, the phenomenon of what you are studying. Alright? Then the next thing you want to look at is the types of bias and error. How many types of bias and error do we have? Okay, so you want to look at the types of bias and error. So if we come here and say types of bias and error, you have different types, up to 8. So I will just list those types of bias and errors for you, and we'll begin to look at how to distinguish them. Alright, so the first type we saw is what? Let me write here, types of bias and error. Alright, so the first one we saw is what? Sampling bias. Number 2, we have measurement bias. So your guess is as good as mine. You can just pretty talk around sampling bias, measurement bias, if you really understand the foundation of bias and error. That's like we have previously looked at right now. And if you are pretty much in class, happy are you, because you could just relate all of it together, because I taught on this in details. So experimental design bias, then we also have data collection errors. Also have participant bias. We also have participant bias. We also have publication bias, confirmation bias, publication bias, confirmation bias. Then we have attrition bias. All right. So these are the types of bias, and of course, from the name, you could easily tell where bias could come from, and with that, you can easily explain. But even your course material that you have can give you clear hints. So each of these bias, you have things like the types, what the type of, for example, for sampling bias you have types and causes. For sampling bias you have, inside sampling bias you have selection bias, volunteer bias, convenience sampling. Okay, for measurement bias you have observer bias, instrumental bias, recall bias. So if you went to school properly, you can even talk around this thing without looking at your course material. You want to measure something like I gave in class, and just when you are supposed to take the last reading, and maybe it's a digital machine connected reading, and light went off, you have failure. You just approximate based on the previous one, and at the end of the day you do not get expected results. Some part of your result is skewed to a wrong direction. That's experimental or measurement bias. Okay, then you so basically for all of them you will see the bias, you will see the types, and you will see the causes. Just generally summarize what could cause each type of bias. Okay, so if I were you, you have sampling bias, you explain what could cause it, what could lead to measurement bias, the types. If there are more than one type, you know them, and you know what could lead to it generally. Okay, that's basically it for all of them, both causes and what types. So thank you. Did I answer your question? Bias and error people, are you okay?
Okay, we'll give you our just keep doing it ourselves.
Okay. Love you too. Come to this thing while you answer me. Come your screen. Okay, so I wish you success in your exams. Try to get some rest this morning, sleep very well. At least with what I've covered, you can be assured of 3 questions for the application of biostatistics to biological research. So if you are able to tackle these questions, they are pretty simple questions, especially if you have been able to attend this examination revision today, because you are kind of more focused and directed instead of reading everywhere and knowing nothing at the end of the day. So that is it, and the essence is for you to pass very well and boost your CGPA, so that if you can now focus on these questions, you could now look at 1 or 2 calculations from the statistical questions. You can, with that, be assured of getting an A in biostatistics, all things being equal. But you know that the practical aspect is solely dependent on biostatistics, so lecturers, so make sure you do what is expected from you. If you need to submit your report, do everything that you need to do to pass very well. I want to thank you for staying till this time. Thank you very much. I like to really know how many persons are here. I can't see that number. I just know the number of views, but I can see concurrently 21 viewers, but it's fine.
No, but not
Okay, so if you dropped your name and all that, I'll see it and I'll know who came, so that who did not come and need help, I also know what to do. Because this is to 3. I cannot stay this long and somebody will still come and fail. Okay, so I'll tell you congratulations in advance. We'll try to put up the other aspect. These lectures is only bias that I did not do a YouTube video, so I just did like a summary now on your request. So but all other videos will be uploaded before morning, so when you rest well and you are doing your final touch, you could look at it. Okay, all things being equal, we'll try to do the same for biotechnology. So hopefully by Sunday into Monday next week, we'll be able to put up biotechnology and genetical engineering to also tackle at least 3 questions from my aspects. Alright, so and it's going to be pretty straightforward and easy. So wish you success. Thank you very much. Bye. Oh, damn, this mosquito chop machine. Industry. Be like all these things. So I be like all those
Totally.